Hello everyone. Discuss blood clotting. Previous class discuss mechanism of blood clotting reactions, the vasoconstriction, platelet plug formation, and blood clotting. So the blood clotting contains three steps. Step 1, formation of prothrombin activator. So completed. Next discuss step 2, conversion of prothrombin to thrombin. Conversion of prothrombin to thrombin. First one, so prothrombin. So prothrombin is a protein. Prothrombin is a protein. It is releasing from the, it is secretion from the liver. So prothrombin is a protein. This protein is releasing from the liver. So this protein is the inactive state. The prothrombin is inactive state. So the first step, prothrombin activator. So the prothrombin activator is conversion of the inactive prothrombin to conversion of the active thrombin. So the liver secretion of the protein prothrombin. The prothrombin is inactive. This inactive prothrombin converting to the active thrombin. Presence of the prothrombin activator and calcium ions. Prothrombin activator and calcium ions. These two, inactive prothrombin is converting to the active thrombin. So these are the step 2 conversion of prothrombin to thrombin. Prothrombin to thrombin. So prothrombin is inactive protein. This is converting to the active thrombin. Presence of the prothrombin activator and calcium ions. Next step, step 3. Next one, step 3. So conversion of fibrinogen to fibrin. Conversion of fibrinogen to fibrin. So these are the fibrinogen. First step, fibrinogen is inactive state. Fibrinogen is the inactive state. So this inactive fibrinogen, presence of the thrombin, presence of the, so these are the active thrombin. This thrombin is activated the fibrin, active fibrin. First step, fibrinogen is the inactive state. So the presence of the thrombin, inactive fibrinogen is converting to the active fibrin. So these are the like monomers. So they are the like these like structures. So these are converting to the fibrin monomers. Fibrin monomers. Next step, these fibrin monomers polymerization. The process, the polymerization. The fibrin monomers, the polymerization process, they formation of the loose fibrin threads. Loose fibrin threads. So they are the one fibrin monomer, so there another fibrin monomer, so there is another fibrin monomer. These are between the fibrin monomers, they formation of the hydrogen bonds, presence of the polymerization. So the fibrin monomers, presence of the polymerization, they formation of the loose fibrin threads. So these threads are called the hydrogen bonds. So the hydrogen bonds are the weak bonds. So they are connecting from the one fibrin monomers to another fibrin monomers. So these loose fibrin threads and factor number 13 and calcium ions. Loose fibrin threads, presence of the factor number 13 and calcium ions. So these are all combined to form the fibrin meshwork. The formation of the fibrin meshwork. So this is like is the fibrin meshwork. So they are connecting from the one monomer to another monomer. Formation of the net-like structure. So the formation of the net-like structure. So presenting of the some gaps. So these are the meshwork, formation of the gaps. So these gaps, these meshwork, the gaps are filling with the blood cells, red blood cells, white blood cells and platelets. The gaps is filling with the red blood cells, white blood cells and platelets. So finally, they formation of the blood clot. Finally, they formation of the blood clot. Next step, these clotting platelets are presenting on the proteins like actin, myosin, thrombocytinin. Actin, myosin, thrombocytinin. These three factors presenting on the blood platelets. So these are the clot retraction. Clot retraction. After blood formation, blood clot formation, after 30 minutes, the platelets contain proteins like actin, myosin and thrombocytinin. These three factors, the formation of the Class retraction, clot retraction. So clot retraction. So the in the same time the blood clotting is the clot retraction. So they swelling out of the yellow color fluid. Yellow color fluid. So this yellow color fluid is called serum. This yellow color fluid is called serum. So these are the blood clotting mechanism. Step 3, conversion of fibrinogen to fibrin. So finally blood clot retraction. So these are helps of the blood platelets containing actin, myosin, thrombosin. These three helps of the clot retraction. So this clot retraction, the releasing of the yellow color fluid. So these are called the serum. Next one, anticoagulants. Anticoagulant. For example, our body presenting on the heparin. So these are heparin is the blood anticoagulant. So our body circulation, blood is clotting? No, because of the presenting on the anticoagulant. So the anticoagulant is the heparin. So blood is does not clot. Next one, the leech. The presenting on the blood anticoagulant, hiridinuria. Hiridine. So they are the one of the blood anticoagulant. Hiridine. So they are the blood anticoagulant. Next one, mosquitoes presenting on the hemolysin. Female Anopheles mosquito presenting on the hemolysin. So they are the one of the blood anticoagulant. So heparin, human blood anticoagulant. Next one, hemolysin. So they are the female Anopheles mosquito blood anticoagulant. Hiridine. So they are the leech blood anticoagulant. For example, in the test tubes, blood test tubes or blood banks. So the blood adding of the citrates or oxalates. Citrates or oxalates adding within the blood. So blood does not clot. So these are the mechanism of clot retractions.